Auzu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Assalamu alaikum, dear students. I hope all of you are fine at your homes and safe. This is my first recorded lecture about soil microbiology. As you know, because of the unprecedented situation of COVID-19, we have to move towards the unconventional methods of giving education to our students. So this system will work like we would be recording our lectures and upload it on some databases or on some servers like YouTube or any other servers. And the links will appear in KCMS. So students can just log into their accounts and then access their the link to this recorded lecture. Along with that, we can also provide recorded, the supplementary material, which student can just go through, and all these things will be helpful to understand the subject material. So what student has to do is to sign in to their accounts and then open the link related to the lecture and also download the material, and then try to understand the topic. In case the student has any doubt or any question, so he or she can just put a question statement on the same KCMS that the lecturer will see and answer to that question in the best possible way. In case there is some problem regarding answering to that query, then we can also wait till the situations are normal, and we can further discuss on other ways. I mean, some other way can be adopted just to clarify that concept. So today my lecture in soil microbiology is about the culturable and non-culturable prokaryotes. As you know, soil is a very complex medium for microbial growth. It contains a huge diversity of populations ranging from prokaryotes to eukaryotes, from bacteria to animals, including plants, fungi, algae, and many other things. When I say it's very complex, it means that the soil is highly variable. Its environment changes just because of its complex structure. So moisture is a very big factor also, because moisture also changes at very small distances. So it means that when we are going to study something in soil, we have to take care of all these things, that we have to cover up the complexity within that. So today's lecture is about prokaryotes, and prokaryotes are something, the simplest type of organisms on this in this. So prokaryotes are of two types, that is the bacteria and archaea. This is a tree you can see here. You can see three types of organisms: bacteria, archaea, and eukarya. This in this tree, it's a very simple tree that is based on small subunit of ribosomal RNA gene. So in this, there are few groups of bacteria that are visible, and same for archaea and eukarya. So in this tree, these two organisms, bacteria and archaea, they belong to prokaryotes. Prokaryotes are the simplest organism on the simplest living organism that are present on Earth. They don't have well-defined membranes. Their nuclear material is directly suspended in a particular place in cytoplasm. So you can see that bacteria and archaea that were supposed to be the same in the near past, before I think before 2000, they have been found as distant from each other as between bacteria and eukarya. So bacteria and archaea are quite distant, and they are distantly placed. They are genetically different. The archaea occur more commonly in the extreme conditions, such as in the salt affected areas, or in the hot springs, or in deep sea environment, etc. So prokaryotes are of different types in soil. For example, they are autotrophic prokaryotes, there are heterotrophic prokaryotes, there are saprophytic prokaryotes. But in soil, when we talk, we mostly talk about the heterotrophic prokaryotes. These are the prokaryotes that can utilize the organic matter for their energy purpose. They depend on the consumption of soil organic matter. They are important part of the decomposer community. Most of the energy flow in soil depends on their heterotrophic activity. Basically, they are important part of all the important nutrient cycles that are they are functioning in the soil, including carbon cycle, nitrogen cycle, including availability of phosphorus in the soil. Similarly, sulfur reactions, and all other things, like when the dead organic matter is added in soil, then these are the prokaryotes which play a very important role in degrading all that organic matter and converting them back into their simpler forms that are either utilized by other organisms or that become available again. So then there is another division in prokaryotes, that is because when we talk about the study of prokaryotes, then we are talking about whether they are culturable or not culturable. The culturable ones are the prokaryotes that can be cultured on plates in the laboratory. The non-culturable prokaryotes are the prokaryotes that cannot be cultured, or they don't appear on the plates. So it doesn't mean that they are not. I mean, they are not heterotrophic. They are also heterotrophic. And another important thing about these two categories, culturable and non-culturable, it is very interesting that only 1% of the soil bacteria are culturable. So we would be discussing all this in our lecture, that why such a small quantity of bacteria are culturable and 99% of bacteria are not culturable. So how do we find that only 1% bacteria are culturable? And how we conclude about that, why most of the bacteria are not culturable? What are those bacteria which are not culturable? What is their contribution towards the uh, soil metabolic reactions, or total soil metabolism? So the culturable soil bacteria are those bacteria which are actually that can be cultured on plates. Uh, Again, as I already talked about that uh, question, why there is such a small percentage of bacteria that is culturable? There are a few important facts about that. Uh, so we can find probably some rationale in that. First is the bias in the methodology. That can be the reason that some bacteria are cultured and we can see them on plates, while others we don't see. For example, we use some culture media in the plates. Again, all the culture media are different from each other, and this is of course a kind of biasness for the bacteria. Some bacteria may better grow in some particular culture media and others may not. Similarly, the incubation conditions. The incubation conditions vary, and they also uh, vary for as far as the bacterial preference is concerned. And then there is a big diversity of bacteria in soil, not only based on morphology but also on physiology. So it's very difficult to pick all of them on plates because sometimes they look very similar and their physiology is slightly different. Then again, the same reason, huge population densities. And the classical taxonomic techniques are very difficult to apply. The classical taxonomy techniques are dependent on some limited characters that are based on either morphology or phenotype or physiology of the bacteria. So similarly, some bacteria grow faster than others. And in this way, they appear on plates, while others we don't see on the plates. So the major classes of bacteria found in soil based on classical taxonomy are the Arthrobacter, that have been found up to 60%, then Bacillus up to 67%, and Pseudomonas, then Agrobacterium, alkali genes, Flavobacterium. So all this composition you see is very variable from one soil to another soil because of big variability within soils, among soils, and also because of the culture media, culture conditions that are too much selective in picking up kind of population out of the, 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 the big diversity present in the soil. The culture conditions are, the media. If you change a media for same soil, you would see different colonies. So media is playing a very important role in selecting which bacteria will be more abundant in a particular test. Similarly, if the soil is coming from forest, or if the soil is coming from grassland, it will contain very huge numbers of different types of bacteria that will be different as compared to the ones coming from a desert or from a culturing from some other type of soil. So soil type is also an important factor because it determines the type of nutrition available to the bacteria, and it also determines the amount of moisture that may be available in the environment. So all these factors affect the type of bacteria that are viable in those conditions. So now we talk about the direct observation of soil bacteria. Different techniques have been used for direct observation of soil bacteria. As we say that only 1% of bacteria are culturable, it means that 99% are not culturable, but we can see them with other methods. What are those methods? Those methods include the light microscopes, fluorescence, or electron microscopes. And these methods revealed that around 100 to 1,000 times higher number of bacteria are visible and are present in the soil as compared to the ones that are viable. Viable means they when you culture, they appear as colonies. And vast numbers of bacteria are extremely small, although they are structurally intact. So such bacteria are so small that their diameter is less than 0.3 microns. Structurally intact means they are viable. I mean, they are living. They are alive. However, because all their structures are okay for starting a life, however, they don't grow, and these are very small cells. And we would call them as dwarf cells in this lecture onward. The methodology in direct observation are different, as you see, and they have different criteria based on which they identify the cell. For example, scanning electron microscopy, SEM, that identifies a cell based on surface and morphology. How is the morphology and how the surface of the cell. And then transmission electron microscopy, that identifies the cell based on the cell membrane or cell wall characteristics. I mean, cell wall or membranes characteristics. Then fluorescence microscopy, that identifies a cell based on a fluorochrome that binds to a certain DNA. So we can see which kind of sequence is present there, and we identify a particular cell. The in the same way, confocal laser scanning, microscopy, and image analysis are very important. And we see that majority of the cells that are visible in direct observations are small. They are dwarf cells, and only 5% of bacteria are larger than cubic micrometer in volume. So, regarding the dwarf cells, the cells, the different properties they have include the first of all, they're very small. The diameter is less than 3 micrometer. Secondly, they are highly abundant in the soil. As you see, more than 95% are the cells which are dwarf. And then they are highly abundant uh, in the soil, and the abundance increases with depth. And then whenever they are tried for growth, they either don't show any growth, or if they show the growth is extremely slow. Their life cycle is extremely slow, so slow that you can say they don't appear on your plates. It seems that these are the cells that have been exposed to drought for long intervals. So we are not, although we are not sure about that, but there are different hypotheses about it. So, secondly, they are stressed by lack of they are probably stressed by the lack of nutrients. As you know, soil is a very difficult medium to grow, and it's sure that this, the communities that are present in the soil, they would be exposed. They will be exposed sooner or later to some kind of stress. The major stress comes when there is a shortage of moisture or there's shortage of nutrients. And nutrients, is something which is limited, and same is true for moisture. So, uh, in one of the hypotheses is that this stress because of lack of moisture or nutrient take them to a point where from where they cannot return back to the normal growth. So the frequency of dwarfs and the nutrients are very much related. I can give you two scenarios as an example. First is the nutrient availability and its relationship with the abundance of small cells. For example, when the depth of soil increase, if we increase the soil depth, we see there is a decrease in availability of nutrients. At the same time, the number of viable cells also decrease, and the number of dwarf cells increase. So we see that with decrease in nutrients, the number of dwarf cells increase. Second scenario is the plant growth. When the plant growth is proceeding towards the end of the season, the nutrient availability decrease, And with the decrease in neutron availability, it has been observed that the number of dwarf cells increase. So there is a very clear relation between nutrient availability and abundance and presence of dwarf cells. While talking about the dwarf cells, if we conclude all our discussion up till now, is that there is a I would say a debate among scientists about the nature of dwarf cells. What are actually dwarf cells? Are they the miniature cells of the otherwise normal cells? Some scientists think that they are actually the stressed cells of the same normal cells. When the normal cells are exposed to nutrient deficiency, as we have already discussed, the size of cells decrease due to asymmetric cell divisions. Others think that they are different from normal cells. They are by themselves. They are not the same as the normal cells are. They have their own physiology. These cells exist by themselves. So, according to this theory, they think that these cells are obligate oligotrophs. Oligotrophs are those populations which prefer low nutrient media, which can only grow in low nutrient medium, like the stressed soil. So they can grow only in low nutrient conditions. And they have their own DNA intact. This is what we have already seen, that when we analyze soil, we see a huge number of communities with their sequences which are uncultured. So because they have their own DNA, because they have their own they are different from the normal ones, so they have their own physiology and they have their own morphology. So this is what this second hypothesis. So if I briefly explain you the dwarf cells here, dwarf cells are the cells which are small in size, and they are the ones which actually don't appear on plates. They are either very slow in growth, or they cannot be cultured simply. You cannot revive their growth. There are different theories about these dwarf cells. One theory is that they are miniature cells of the normal cells that we see on plates. So because of stress conditions, either because of nutrient or moisture stress, their size shrink and they become small. This is one set of theory. The second one is that these cells are different from the normal cells. They are basically normal obligate oligotrophs. They prefer conditions which are low in nutrients, and they have their DNA intact. They have their own physiology and morphology, and they have high surface to volume ratios. What does it mean, surface to volume ratio? It means because their size is small, they have bigger surface to the exposed to the environment, so that they can take advantage of the bigger amount of possible absorptions from the outer environment. So here I explain you a little bit about a method that can be used, or that has been used, to separate bacteria in the soil based on their size. This method was based on the blending and separation of cells from the soil colloids, based on centrifugation and then uh, separation, in which supernatrants are picked, and then again density gradient concentration, which separate the things based on their densities, followed by filtration with different pore size. Different pore sizes allow to separate the different size cells. So now we have discussed about the dwarfs and the culturables. We have already compared, but what are the contribution towards the overall soil turnover in terms of biomass, or in terms of their physiology or metabolism? So this is what I would like to explain here. If we see here in soil, the number of dwarfs is huge as compared to the culturable. The volume of dwarfs, however, is only 10% of the total biovolume. So this is very important. So although more than 95% are dwarfs, but they only contribute 10% of the total biovolume. Then the dwarf cells account for only minor fraction of the total energy flux of the soil bacterial communities. This is another very important. So they don't have major contribution in the energy flux of the soil, although they are genetically intact. So this is what we can extract out of here, is that although they are genetically intact, but they have negligible physiological significance in the soil microbial communities, and there is a inverse relationship between cell size and cell turnover. So if we summarize all what we have discussed till now, is that there is a huge population of bacteria in the soil, which are small in size, and we call them dwarf. Each dwarf community is basically not culturable. The culturable bacteria, they account for practically all the growth and physiology of the soil communities. The non-culturable bacteria are of minimal metabolic importance in soil, although they are in big numbers. The DNA information of non-culturable communities is overwhelming, as the DNA information are intact. So now we move on to next. So now we discuss about the starvation response of bacteria in the soil. So as you know, soil, if the bacteria is living in soil, it should be exposed to some kind of stress because of starvation due to lack of nutrient, or because of lack of moisture, they're exposed to some kind of drought. So whenever a bacteria starts its growth in soil, it will grow until either it is stopped by some kind of predator, or it is stopped by resources. Where as soon as the resources are vanished, as soon as the resources are finished, they will stop. Their growth will stop, or they will encounter some kind of predator, and the predator will reduce their population size to a limit where they will continue to grow as normal. So because in soil the bacteria are living in a very sophisticated kind of environment in small pores, mostly predators have difficult uh, access to these bacteria. Therefore, normally they encounter some kind of stress. So, and the soil-borne bacteria have ability to withstand the stress. Whenever they face some kind of stress, there is a reduction in the ribosomal RNA that shrinks. Then the proteins are degraded, some kind of contents of proteins are degraded. Then we see a series of genes are expressed, that these genes which are expressed in stress are known as starvation specific genes. So these genes actually increase stress tolerance. Then because of the gene expression, stress induced proteins are released, and then these bacteria can produce some kind of resting stages. If they can't, they are just die off. And it has been observed that only small fractions of resting stages can regain their growth upon onset of the normal conditions. So from here, we get the concept of dormancy. So dormancy means all the things are intact. The bacterial stage can regain its growth, but at the moment they are dormant. Their response doesn't start. The second is a condition where the starvation may take the cells to a stage where they cannot return to the normal growth. This we can say that it's a kind of death of the cell. So the two concepts are there: concept of dormancy and then concept of death in bacteria in the soil. So when we talk about the starvation response, some bacteria such as bacilli and clostridia, they produce internal spores that are known as endospores, as you can see in this figure. These endospores are highly resistant to extreme conditions like extreme temperature, radiation, pressure, and environmental stresses. They are the most resistant biological structures known. They are responsible for the persistence and survival of bacteria for many years, and they regain their growth when the normal conditions are on. Now, what are the methods, molecular methods that can be used to study the communities in the soil, especially bacterial communities? The molecular methods are mostly based on 16S RNA gene, which is used as a universal barcode for detection of bacteria. In this case, the whole DNA from the soil is extracted from the soil. Means all the DNA of all the organisms are extracted. And then we use specific primers by PCR specific primers that can amplify specific regions in DNA that are specific to the bacteria or prokaryotic region. Then we can clone and sequence the amplified regions. The drawback of this technique is that, again, we extract, when we extract DNA, again those bacteria which are bigger in size, they have more probability to, to, to for extraction. Then they may be extracted better than the dwarf cells. Smaller the size, lesser is the cell lysis by different techniques. So ideal way is always to separate the cells based on size, as I have already discussed earlier in this lecture. So we can separate the sizes and then we can perform the extraction of community DNA. So so far, by different molecular methods, by different molecular studies, we have observed that that there is a big number of communities of bacteria or prokaryotes which have no representative or very few representative as cultured organism. So they have no cultivated representative. So this technique also support the idea that the huge number of bacteria in the soil that have their own DNA, they cannot be cultured. So these bacteria or these groups have high abundance in many soil environments, and we see tremendous diversity within the taxonomic groups that were earlier established on the basis of cultivated organisms. And we see new groups, new subgroups within these established groups. So here, this chart gives you a small picture in which you can see the divisions of different groups, and each group contain non-cultured organisms. Very few cultivated examples are there. Now, there is another important group that I have separated to discuss here while I'm talking about prokaryotes in soil. So we should discuss about specifically about thread-like bacteria in soil, which are just like fungus, but they are not fungus basically. They are bacteria, they are single-celled, but they make colonies, thread-like colonies. When they divide, the cells remain attached. They are basically actinomycetes. As I have already explained, they are much more abundant in the soil. One species is example of one species. One example of species is Arthrobacter. Up to 67% abundance we see by this species, that is Arthrobacter species. Another type of bacteria that make chains are cyanobacteria. They make chains of cells along with heterocysts. Heterocysts, you may study about it. Heterocyst is actually a cell that is non-autotrophic. It's heterotrophic. but it gets its energy, its photosynthate, or its carbon from from the other cells, which are actually autotrophic. Cysts are there in the colonies to fix nitrogen. So at the end, I would share the summary or take away points of this lecture, that culture bacteria are very small in numbers, but contribute most to the soil physiology and metabolism, first of all. Then the non-culturable bacteria are huge in number, but they are small in size. And we can say that they are small in size, so they are also known as the dwarf cells. However, they have a very small contribution towards physiology and metabolism of soil. There are different hypotheses to explain the non-culturable bacteria in the soil communities. One I already discussed in this lecture is that some people think that they are actually stressed cells of the same normal communities that we see in the culture form. Second community say that no, they are actually the cells of their own. They have their DNA intact, and whenever they are studied using molecular techniques, they show their their own groups, as we have already seen in this lecture. Then we discussed about the concept of dormancy and death. It's different from normal, as we have the concept in our normal life. In soil, we see number of cells which are dormant, and we expect them to resume their growth after a fixed number of years or fixed period of time. And there are cells we think that they may not resume their growth, so they are actually dead cells. Molecular studies have reflected huge numbers of non-culturable bacteria, along with the small numbers of culturable bacteria. The drawbacks of molecular techniques are also there, as we discussed in this lecture, that the extraction techniques are a bit biased or selective towards those who are bigger in size. Thanks very much.